Hello everyone. In the previous 3 videos on biochemistry of metabolism, we have studied basic overview of metabolism, integration of metabolism at cellular and organ level, and properties of metabolic pathways. We all know that our body is like a community of interacting tissues and cells. Individual cells do not work in isolation. So how do these cells communicate with each other? So let's find out the answer in this video on signaling pathways of metabolic regulation. A sophisticated communication system exists in the body which coordinate the functions of all the cells through signals. And these signals can be intracellular, intercellular, or through second messengers. In the carbohydrate, lipids, and protein metabolism, the most important route of communication is chemical signaling through hormones. And insulin, glucagon, and epinephrine are the major hormones which play a key role in metabolism of these biomolecules. And these three hormones act through second messenger system. These are the group 2 hormones which act on cell surface receptors. In the second messenger system, hormone acts as a signal, the receptor acts as a detector, and enzyme acts as an effector. So when hormone binds to the receptor, that complex initiates series of reactions that ultimately result in a specific type of intracellular response which is mediated through the second messenger. And why it is called second messenger? Because the first messenger is hormone itself and it intervenes between that first messenger and effect on the cell. There are three major signaling pathways in fuel metabolism. The first one involves adenylyl cyclase, and cyclic AMP is second messenger in this pathway. The second pathway involves calcium and phosphatidylinositol. Calcium inositol triphosphate and diacylglycerol are second messenger in this pathway. The third pathway involves receptors with intrinsic tyrosine kinase activity. The hormones like epinephrine and glucagon act through adenylyl cyclase pathway. Epinephrine can also act through calcium phosphatidylinositol pathway but through alpha adrenergic receptors. And insulin acts through receptors with intrinsic tyrosine kinase activity. And both the adenylyl cyclase and calcium phosphatidylinositol, both the pathway acts through G protein coupled receptors. These G protein coupled receptors are also called as 7 transmembrane receptor or serpentine receptors. It has one extracellular domain for binding with the hormone, 7 transmembrane domain, and it has one intracellular domain as well which interacts with G protein. And when hormone is bound to this receptor, it communicates with effector through the G protein. So it communicates with the effector for the further action through G protein, and that's why it is called as G protein coupled receptors. This G protein is a heterotrimeric protein and it has 3 subunits, alpha, beta, and gamma. So when the receptor is not bound by hormone, this G protein remains inactive and separate from the receptors. And when the hormone is bound to this receptor, there is transmission of signal to this G protein, and this GDP is exchanged with the GTP, and this G protein will ultimately activate the effector. And the G protein which is involved in this pathway is called as GS protein. And the effector which is involved in the adenylyl cyclase pathway is adenylyl cyclase itself. And through this adenylyl cyclase, there is formation of cyclic AMP, which is a second messenger in this pathway. Now what happens in the calcium phosphatidylinositol? So epinephrine acts through alpha adrenergic receptor. So this 7 transmembrane receptor or this GPCR is a alpha adrenergic receptor in this pathway. And the G protein which is involved here is a GQ protein. And the effector here is phospholipase C. So through this phospholipase C, this is activated by this GQ protein, and there will be formation of second messengers like inositol triphosphate and diacylglycerol. So these are the 3 important signaling pathways in fuel metabolism. Insulin is a hormone which is secreted by beta cells of pancreas, and it is a hypoglycemic hormone. It acts through The receptor having intrinsic tyrosine kinase activity. So let's see how it works. Insulin receptor has two types of subunit, alpha and beta. The intracellular domain of beta subunit has intrinsic tyrosine kinase activity. So when hormone insulin binds to alpha subunit, the signal is transmitted to beta subunit and the tyrosine gets phosphorylated. Now this phosphorylated tyrosine kinase phosphorylates other insulin receptor substrates. And this also gets phosphorylated and activate various signaling pathways like PI3 kinase, mTOR, GRB2, mSOS. We won't go into details of these pathways in this video, but ultimately insulin results into its biological actions like protein translocation, that is glucose transporter and insulin receptor, and enzyme activity. It alters enzyme activity like enzymes protein phosphatase, phosphodiesterase. It also acts on the gene level by increasing or decreasing transcription of certain enzymes like glucokinase, phosphoenol pyruvate carboxykinase. Insulin increases glucose uptake in the cells like muscle and adipose tissue through GLUT4 receptors, and it increases glycogenesis, protein synthesis and fat synthesis, and it decreases gluconeogenesis, glycogenolysis and lipolysis. So insulin is a hormone which is secreted by beta cells of pancreas in the well-fed condition, and it regulates blood sugar level, and it is a hypoglycemic hormone. Now let's see action of glucagon and epinephrine through adenylyl cyclase system, which involves cyclic AMP as second messenger, and both the glucagon and epinephrine increase cyclic AMP level. Imagine this is a cell, this is cell cytosol, this is plasma membrane, this is G protein coupled receptor, and this is trimeric G protein having alpha, beta and gamma subunit. When this receptor is not bound to hormone, this G protein is separate from this receptor and alpha subunit is bound to GDP. This is adenylyl cyclase, it is effector. This is inactive protein kinase A having two regulatory subunit and two catalytic unit. When hormone binds to this receptor, it undergoes conformational change and signal is transmitted to G protein. So this GDP then converts into GTP, and this alpha subunit along with this GTP, it dissociates from beta and gamma subunit and it goes to adenylyl cyclase and it activates adenylyl cyclase. But this alpha subunit has intrinsic GTPase activity, so its action is short-lived. And GTP is again hydrolyzed to GDP. Because of this hydrolysis, this alpha subunit gets dissociated from the adenylyl cyclase and it goes back to its original position and gets reassociated with beta and gamma subunit. Now what is the function of this activated adenylyl cyclase? It converts ATP into cyclic AMP. Cyclic AMP is a second messenger. It binds to regulatory unit of inactive protein kinase A, and because of this catalytic unit are released. Now what is the function of this catalytic unit? This becomes now active protein kinase A. So it catalyzes the transfer of phosphate from ATP and it transfers that phosphate group to this protein. So now this protein becomes phosphoprotein. So for example, if this protein is enzyme, then by the action of this active protein kinase A, that enzyme becomes phosphorylated enzyme. And this process is called as phosphorylation. Now how this protein again converted into protein? By dephosphorylation by the action of enzyme protein phosphatase. So phosphorylation and dephosphorylation are reversible covalent modifications, and this is one of the important mechanism of regulation of enzyme activity. Now once the cyclic AMP activity is over and hormone is not associated with the receptor, this cyclic AMP is converted into 5' AMP by the action of enzyme phosphodiesterase. Now let's see how insulin opposes the action of glucagon and epinephrine. Insulin activates this enzyme protein phosphatase. So what is the action of insulin? it converts phosphorylated enzyme into dephosphorylated enzyme. So insulin causes dephosphorylation while glucagon and epinephrine causes phosphorylation. In addition to this dephosphorylation action of insulin, what other action insulin performs? It activates the enzyme phosphodiesterase, which is responsible for conversion of cyclic AMP into 5'-AMP, and by doing this it decreases the concentration of cyclic AMP, so which in turn opposes the action of glucagon and epinephrine. The enzyme glycogen phosphorylase and hormone sensitive lipase, the activity of these enzymes are increased by glucagon and epinephrine. It means that these two enzymes are active in phosphorylated condition. So glycogen phosphorylase is an enzyme of glycogenolysis and hormone sensitive lipase is a key enzyme of lipolysis. So both these mechanisms, glycogenolysis and lipolysis, are activated by glucagon and epinephrine, and these two enzymes are active in the phosphorylated condition. Glucagon and epinephrine decreases the activity of glycogen synthase, which is a key enzyme in glycogenesis. Acetyl-CoA carboxylase, it is a regulatory enzyme in fatty acid synthesis. HMG-CoA reductase, it is a rate-limiting enzyme in cholesterol synthesis. So both glucagon and epinephrine, they decreases the activity of all these enzymes. It means that these enzymes are active in dephosphorylated condition. So this is the mechanism of action of adenylyl cyclase system, in which the cyclic AMP is a second messenger. Now let's see how epinephrine acts through calcium and phosphatidylinositol pathway. This is a schematic representation of a cell showing cytosol, cell membrane. This is endoplasmic reticulum having calcium reservoir in it. This is a calcium channel. This is G protein coupled receptor associated with G protein having alpha, beta and gamma subunit. This is phosphatidylinositol bisphosphate. This is an effector phospholipase C, and this is a calcium channel. When hormone like epinephrine binds to this G protein coupled receptor, that is alpha adrenergic receptor, there is conformational change in the receptor. The GDP, which is bound to alpha subunit, converted into GTP, and it gets dissociated from beta and gamma subunit and goes to the effector. It activates the effector, that is phospholipase C. But this alpha subunit has intrinsic GTPase activity, so its action is short-lived, and GTP is again hydrolyzed to GDP and gets dissociated from the effector, that is phospholipase C, and it goes back to its original position and reassociate with the beta and gamma subunit. Now this phospholipase C is activated, so let's see its action. The phospholipase C causes the breakdown of phosphatidylinositol bisphosphate into diacylglycerol and inositol triphosphate. What is the action of this inositol triphosphate? It goes and binds to calcium channel. It causes exit of this calcium from interior of endoplasmic reticulum into cytosol. In addition to this, G protein directly can activate the entry of calcium from exterior of cell to the interior of cell. This in turn leads to increased concentration of calcium inside the cell. And the protein camodulin binds to calcium to form calcium camodulin complex, which activates camodulin kinase. What is the action of diacylglycerol? Diacylglycerol activates the enzyme protein kinase C. The activated protein kinase C and camodulin kinase will phosphorylate the enzyme, and this phosphorylate enzyme is active form of enzyme, and this pathway is very important in glycogenolysis. There are various enzymes which gets activated by this pathway, but most importantly phosphorylase kinase and pyruvate carboxylase. Having learned about the three signaling pathways of insulin, glucagon and epinephrine, let's now discuss the effects of these hormones on fuel metabolism in brief. Let's first see the action of insulin. Insulin is a hormone which is secreted by beta cells of pancreas, and when it is secreted in the well-fed condition, whenever there is increased concentration of glucose in the circulation. The GLUT2 receptors, that is glucose transporter, which are located on the liver, they are not insulin dependent, they are insulin independent. So in case of increased concentration of glucose, it is taken inside the liver by GLUT2 receptor. And insulin, now what is the action of insulin on liver? It binds to the insulin receptors present on the liver. The receptors gets activated, and through the series of reaction the response is generated. And what is the response? The excess glucose is stored at glycogen. So insulin favors the process of glycogenesis. In addition to this, in liver, insulin favors the synthesis of lipogenesis. Okay, now on the muscle, what is the action on muscle? In the muscle, insulin receptors are located, and the GLUT4 which are present in the muscle, they are insulin dependent. So whenever there is hyperglycemia, this insulin goes and binds to this insulin receptors present on the muscle, and it gets activated, and it brings this GLUT4 receptor to the surface. Okay, so there is translocation of this GLUT4 transporters to the surface so that the glucose can be uptaken by the muscle cell. So these GLUT4 receptors present in the muscle are insulin dependent. Now what is the action of insulin here in muscle? The excess of glucose is also stored as glycogen in the muscle. So glycogenesis is favored in the muscle by insulin. Now let's see the action of insulin in the adipose tissue. Adipose tissue also has GLUT4 receptors which are located in the intracellular vesicles, and they are insulin dependent. So when insulin binds to the receptors, they cause transport of this intracellular receptors to the surface, and through which glucose can enter inside the cell. And what is the action of insulin in adipose tissue? It causes lipogenesis, that is fatty acid synthesis and then storage of fatty acid in the form of triacylglycerol in adipose tissue. So these are the various actions of insulin in the various organs like liver, muscle and adipose tissue. What are the effects of glucagon and epinephrine? Glucagon is synthesized by alpha cells of pancreas in the fasting condition whenever there is decreased concentration of glucose. And epinephrine, which is also called as adrenaline, it is synthesized from adrenal medulla, and it is synthesized in the condition of stress whenever there is anticipation of muscular activity in case of fright and flight. So let's see what are the actions of these two hormones on various organs. The glucagon binds to glucagon receptor which are present on the liver, and it activates the glucagon receptor. And liver is more responsive to the action of glucagon than epinephrine. The action of epinephrine on liver is not that as significant as the action of glucagon. But the epinephrine also binds to the adrenergic receptor, which is beta adrenergic receptor, and both causes glycogenolysis. So breakdown of glycogen into glucose and synthesis of new glucose. So the glucose which is synthesized in the liver then transported to the tissue which require this glucose in fasting and starvation, and it goes into circulation through GLUT2 receptors. Now let's see what is the action of this glucagon on adipose tissue. So this glucagon binds to glucagon receptor, activates that. The adrenaline or epinephrine also goes and binds to beta adrenergic receptors, and together they activate the process of lipolysis, that is breakdown of TAG, triacylglycerol, and fatty acids are released in the circulation which are utilized by muscle, liver etc. Brain cannot utilize fatty acids. Now, what is the action of this epinephrine on muscle? This muscle is unresponsive or insensitive to the action of glucagon. Only epinephrine causes glycogenolysis in the muscle. These adrenergic receptors are responsive to epinephrine. The hormone binds to the receptors, it gets activated and it causes glycogenolysis. And glucose 6-phosphate is released or formed, but it cannot be further converted into glucose because the key enzyme, that is glucose 6-phosphatase, is absent in the muscle, and that's why that glucose 6-phosphate is utilized by the process of glycolysis in muscle. And that's why muscle glycogenolysis is not doesn't play any role in the maintenance of blood glucose concentration. So this was about the action of glucagon and epinephrine on different types of organs. Let's summarize today's session. There are three major signaling pathways of metabolic regulation. The first one involves adenylyl cyclase, and cyclic AMP is a second messenger in that pathway. Glucagon and epinephrine act through that pathway. The second is calcium phosphatidylinositol pathway, wherein calcium inositol triphosphate and diacylglycerol are second messenger. Epinephrine acts through this pathway involving alpha adrenergic receptor. The third pathway involves receptors with intrinsic tyrosine kinase activity, and insulin acts through this pathway. We have also discussed in brief the metabolic actions of epinephrine, glucagon and insulin. In the next video on the metabolism series, we will discuss about the regulation of metabolic pathways. If you find this video useful, please like, share and subscribe and write your queries in the comment section.